is Anna Glutley, and I am going to demonstrate how you should read a biology ebook and take notes. There's a tendency for students to want to read a textbook on their iPad or their computer screen the same way they would a Facebook page or an internet website. And there's a different technique to it. You need to kind of change your mindset and start thinking about your digital books, your ebooks, more like you would a paper book. So pretend that you're reading a paper book, okay? So I'm going to change color here. So pretend. Pretend that it's a paper book, okay? It's really important that you not do a couple of things that people typically do when they're reading a website, which is to skim, all right? You don't want to skim, okay? And you don't want to read it like Facebook or the internet, okay? These are all big no-nos, okay? So instead, what you're going to do is you're going to read it like it's a paper book, okay? And when you're reading, I want you to be mindful about what you're reading and think about it, okay? This is going to force you to go slow, okay, and put effort into what you're reading, okay? Now, as you're doing this, realize that you're reading on a digital screen. The lighting is different. A dark background versus a light background. These can all affect your eyes. So you want to be aware of eye fatigue. Take whatever app you're using, whether it's iBooks or Kindle or a PDF reader, and change it so that it is not hard on your eyes. All right. And remember to take frequent eye breaks, which basically means look away from your screen, look into the distance, and then look back at the screen. Okay. Now, as you are being mindful and going slowly, I want you to annotate what you're reading, okay? Annotate is the super fancy word for take notes, okay? And then when you're done with your reading session, this is a really helpful tool, which is to reflect on what you've read. So what did you read? You can write it down, or you can just do this in your head. So basically kind of summarize for yourself what you were reading about, okay? Think about how you were reading it. When you're done reading, did you read it fast? Did you read it slow? Did you take notes? Do you feel like it was a successful session, okay? And then think about why you're reading this. Are you reading it for fun? Are you reading it because you need to study for an exam? Or are you reading this in preparation for writing a paper? These all change how you approach your reading, okay? The important point all of this is to think about your mindset as you read and how you approach your reading, okay? So I'm going to model how I would read a science textbook, all right? And so here's the scenario. I'm going to be using an OER book from OpenStax. This is a free textbook. It's the Concepts in Biology, okay? And because I'm on an iPad and it's easy for me, I'm going to open up this book in iBooks, okay? And with OpenStax, you can either do this in PDF format, or they have an iBook format, which you have to pay for. The PDF format's free, okay? The iBook format isn't, but you can open either of them in iBooks. I could have done this in Kindle, or I could do this in PDF Viewer or Foxit, or if I was using a publisher's book, I might be using an app that's been put out by the publisher, whether it's McGraw-Hill or Pearson, okay, or Wiley or one of the others, okay? And then, really important, when you are reading, especially for a class, you have to do note-taking as you are reading, okay? Now, in this case, I'm going to be using the app Noteability, okay? Now, you could use a different app. You could be using Word, You could also just be doing your note taking on paper. But what I want to do is, as I'm going along, I want to be annotating by doing highlights and underlining or circling, depending on what kind of app you're using, within the book that I'm using. And then I want to be writing extra notes in another app or on a piece of paper. Okay, so I'm going to continue doing that right on this page, and I'm going to scroll this up, and I'm going to change my screen. I happen to be, I guess I'll put what tool I'm using. I'm using an iPad Pro with the Apple Pencil. And again, you don't have to use these. You could be using something else. You could be using an Android tablet with a different type of stylus. You could be using your phone and then using a piece of paper to write notes on. A lot of different options, okay? Now I'm going to change the screen, all right? Okay, so now I am in the OpenStax book, all right? If I tap up here, I can see that there's a table of contents. I'm just going to stay where I'm at, and I want to kind of demonstrate how I'm going to be doing this, okay? So I am in the iBooks, and I'm going to come down here, and I see a prokaryotic cell is a simple single-celled unicellular organism. So this is highlighted, so I know that this is important, okay? And because of iBooks, I can do that, but I don't need to do that, okay? So what's important, it's single-celled, all right, lacks a nucleus, it doesn't have any membrane-bound organelles, so that's important. It's found in the central, in a darkened area called the nucleoid, okay? So I know that I need to probably be able to review this. Now, these highlights I can always go back to, but there's a tendency when you're reading the notes that you took originally for you to just kind of go through and not really think about what you're doing when you're reviewing something for a test. So it's a lot more useful if you actually go and write things down, all right? So I'm going to write my notes. So I know that over here I've got a prokaryote, okay? And what are the important components of the prokaryote? I don't want to read this whole paragraph when I'm studying for a test. I just want the key points. Well, it's a single cell. That's really important, all right? And then there's no nucleus and no membrane bound organelles, okay? And the DNA, all right, is located in the nucleoid, all right? Oh, geez, and I don't know what a nucleoid is, so I'm just going to draw a little arrow over here to define, okay? And the nucleoid, if I'm over here, so that's a darkened region. Okay, so that's a darkened region of the cell, okay? And it tends to be in the center. Okay, so now I'm going to come over to the other paragraph. Okay, so I've got, unlike archaea, eukaryotes, bacteria have a cell wall made of peptidoglycans composed of sugar and amino acids, and many have a polysaccharide capsule. Okay, so if we come back up to this paragraph, I guess I should have started up here. So the predominantly single-celled organisms of the domains bacteria and archaea are classified as prokaryotes. Oh, that's an important point. So the prokaryotes include bacteria and archaea. Okay, so that's an important point. Now it mentions up here the eukaryotes, and I know that that's going to be something different. So I'm going to just kind of remember that, and then I'm going to not do too much with that until I get to the section on prokaryotes. Now, right here in this section, well, they've got this before and this nucleus. So that's telling me that there's something important about the name. This is a clue. So I've got pro, all right? And pro means before. And then karyote means nucleus. Okay. So you don't have to, when you're approaching a page, you don't necessarily have to read everything in order. All right. You can start with a paragraph in the middle or at the end. Often it makes more sense to start at the beginning, but you can see what I did there. Okay. And I just went back because I was like, oh, wait a minute, I missed some information. That's important for you to do as well. If you've missed something, go back to it and reread it and be like, oh, I need to take notes on that. Okay. So this coming down to this last paragraph again. So unlike archaea and eukaryotes, bacteria have a cell wall made of peptidoglycans. Okay. So archaea don't have, it's not having a cell wall. So I'm going to go back to my black for my main notes, all right? And I'm going to say bacteria, okay? I know they're a prokaryote, all right? Plus has a cell wall, okay? So I'm going to just put that in there. It's an extra layer of protection. I haven't decided if I want to take notes on that yet. It might be an important detail, but I can always come back to that. Okay, so it's helping it maintain shape and prevent dehydration. Some prokaryotes have flagella, pili, or fimbriae. Okay, so a feature of prokaryotes, okay, they have flagella, pili, or fimbriae. I'm not real sure what all those things are. So I know that those are all things that I'm going to underline because I need to look them up and make sure I include the definitions if I don't know what they are. So right here, flagella are used for locomotion. So I'm going to add that note in there, locomotion. Okay, pili are used for, let's go to the next page, to exchange genetic material. So I can't quite visualize what pili are, but I'm just going to say what the function is, which is to exchange genes, okay? And it does that in conjunction, okay? So conjunction, again, not a word that I might remember, so maybe I need to kind of circle that to remind myself that I need to go and define that in my notes, okay? And then fimbriae are protein appendages used to attach to other cells. Okay, so it's like an arm. So appendage is like an arm that attaches bacteria to other cells. Okay? All right, so now we're getting up into eukaryote. And I can see, well, I've already gone through and done a little bit of highlighting. All right, which is kind of nice to know. So, you've read it through it once. So in nature, the relationship between form and function is apparent at all levels. All right, the principles form follow function. So they put that in quotation marks. So that probably is important. Birds and fish have streamlined bodies. In general, one can deduce the function of a structure by looking at its form. Okay, so there's a lot of information in that paragraph, but most of it isn't really important. It's explanatory. It's helping you understand the concept, but it's not necessarily something you need to rewrite. But something that is important is what the concept was. So form follows function. Okay, and if you can remember that, what that means, you don't need to write down anything else. But if you can't remember that, then you would come back over to your highlights where it says deduce the function of a structure by looking at its form. Okay, so that's right here. Okay, and you might write that down in your notes, but if you can remember it, then you don't need to. Okay, and then we're coming over here. A eukaryotic cell is, okay, so I know a cell that has a membrane-bound nucleus. So I'm over here, and I'm going to write down eukaryote. Now, this is important because basically what they're doing is they're telling you how a eukaryote is different from a prokaryote. So these are the really important points. If you come back up here, you'll see that you wrote single cell, no nucleus, no membrane-bound organelles, DNA is in the nucleoid, okay? Now what you're going to do with the eukaryote is write what that is. So a cell that has a membrane nucleus. So you have a proper nucleus with a membrane around it. Okay? You have other organelles with membranes. Okay? And then you have a, okay. And then right here, so we've got true kernel, all right? So if you remember we were defining prokaryote, so U is telling you that means true, and then karyote is telling you that that is the kernel or nucleus, okay? So that's the information that you're doing, that you're putting it all together. Now, what's an organelle? So I've got the word organelle, and it means little organ. So right here, I'm going to keep with my blue for definitions, and this means little organ. Okay? They all have specialized cellular functions. So I'm going to put that over here. Okay. And then the next section, it's going to start talking about cell size. And I'm not actually going to write any notes right there because I don't want to make this video too long. Okay. So when you're done taking notes, you want to do a reflection on your notes. Okay. And you can do this again in your head, or you can write it out on paper. I like writing out reflections, and this doesn't need to take a lot of time. I'm thinking 1 minute. Okay, and this is where you would basically say that, I read about, all right, and then you would explain what you read about. So we read about the eukaryotic and prokaryotic cells. And then how did you take your notes? At this point, you would talk about how you read slowly and you were annotating and highlighting. And you might put in a clarifying point, like you're taking your main ideas in black color, and then you're using blue for definitions, that kind of thing. So that way you can go back in case you've forgotten why you've done your notes in a certain way, and you can figure that out. Okay, so the conclusion to this video, all right, so the purpose of this video was for me to model an ebook. Okay, so I'm modeling how to read a biology ebook slowly while annotating and taking notes. Okay, now what I really want you to get out of this is that I was going really slow, all right, and I was really thinking about what I was doing, and I was trying to take notes on what I was doing. I wasn't going through and quickly highlighting as I read. I understood what I read because I'm taking notes and I'm trying to organize the information, get rid of all the extra words in the paragraphs, and write down the details that I need in order to study for a test, in order to understand a concept, or to be able to gather the information I need in order to write a paper. So you need to do this when you're reading an ebook, all right? And the skills that you use for reading an ebook with the way that I've modeled, this is actually how you would read a paper textbook as well. It is the same technique, whether it's a paper textbook or an ebook. The important thing is to remember that your ebook is a book, not a website. All right, so that is the end of this tutorial on how to read a biology ebook and take notes.